The overall goal of this analytical protocol is to simultaneously extract and quantitate the two anti-cancer drugs, Doxorubicin and Mitomycin C, co-delivered by polymer lipid hybrid nanoparticles, and the doxorubicin's metabolite, doxorubicinol, in biological matrices, using a simple and robust reversed-phase high-performance liquid chromatography method.
This method can help answer key questions in the field of nanomedicine, such as how to design an effective nanocarrier system based on the nanopharmacokinetics of drug combinations. The main advantage of this technique is that it allows for the simultaneous quantitation of 3 drug compounds in the same biological matrix without the need to change the mobile phase of HPLC.
Though this method can provide insight into the biological behaviors of doxorubicin, Mitomycin C, and doxorubicinol in primary breast tumors, it can also be applied to other cancer types treated with similar drugs. Visual demonstration of this method is critical, as efficient sample preparation steps are difficult to learn.
To begin this procedure, collect and freeze the whole blood, major organs, and breast tumor as outlined in the text protocol. Quickly weigh the frozen tissues and record the weight on the lab notebook. Then, transfer them into a 13 milliliter round-bottom conical tube. Next, add between 1 and 5 milliliters of ice-cold cell lysis buffer. Using an electric hand homogenizer, homogenize the tissue with an up and down stroke motion on ice at 18,000 RPM. After this,